So we have Biological Psychology, 2nd edition, 1999, MIT Press, Rosenzweig, Lyman, and Breedlove. And in box 4.2 here on chapter 87, I have a discussion here of the NMDA and AMPA receptors in the dendrites. And that is the N-methyl-D aspartate and the alpha-amino 3-hydroxy-5-methyl-4 isoxazole propionic acid receptors. And those are special because when we learn, electricity causes these to go. There's this chemical that flows, this calmodium, calcium calmodulin, Ca2 plus ion, and it comes into this NMDA receptor here. And not very much of it can get through. It's glutamate is the chemical. And it can't get in very well because there's this barrier, this magnesium barrier there between -75 and -35 millivolts. But when AMPA is also present, then the barrier is removed. The membrane, it depolarizes, and lots of this Ca2 plus ion, this calmodulin, calcium calmodulin protein kinase can enter the cell. And what that does, we're going to go over and see my lovely bookmark made out of a paper towel over here to this part. And so you have this stuff called long-term potentiation, and that's a measure of electrical charge in cells after a charge is applied. And you have like pre-synaptic potential and post-training potentials, and these different things that are used to describe learning. And so we put little electrodes in rat brains and put them through mazes and then zap them and listen to them with EEGs and look at what happened to their cells, and that's how we can determine whether or not they're learning. That's how we understood this. So, and then a lot of microscopes. So we have the AMPA and the NMDA receptors here, and I'm going to go through chapter 18 here. This is the biological process of learning, memory, and neural mechanisms. So, in the guise of Patrick Stewart, AMPA and NMDA at the dendritic spire, where the axon terminal does provide glutamate, and the calmodulin Ca2 plus ions do flow, and the magnesium membrane does depolarize, and allow them to go forth into the cell. And therein pKa, pKc, and CAM do release proteins, which activate kinases to bind to calmodulin response-based proteins, which lead to the creation of messenger RNA to create immediate early genes, which in turn are taken from the endoplasmic reticulum that are used to create late effector genes to form new cells and new pathways in the brain. Literally, learning is evolution.